Katie Irving is going to talk about Arroyo toad and some of its vulnerabilities to hydrologic change. Let's go for it, Katie.
Thank you very much. Hi everyone. So I'm Katie Irving. I'm a scientist at SCWRP. I think I'm the third SCWRPer in a row, but I think I'm also the last. So I'm going to talk today about the Arroyo toad and how we modeled it in terms of vulnerability to hydrologic change in the San Diego region. So just a bit of context, flow alteration is pervasive spatially, but it's also a concern at multiple timescales. So by that, I basically mean, so contemporary alteration is stuff that's already happening, and it's already like the current situation. So modifications and diversions and things like that is what is impacting current, is what's going on currently. So the LA River is a pretty obvious example of that. And then near term is basically what's going to happen in the kind of more immediate future. So I don't want to put a time frame on it, but like 10, 20 years or so. So any changes to water rights or any other diversions from other projects are going to impact the alteration in the near term. And then long term is pretty much climate change. So how increases in temperature and differences in precipitation are going to impact flow. So with that in mind, it's really a good idea to understand the vulnerability of streams in the long term, because that's going to impact decisions that are made in the near term, right? So understanding the vulnerability, this is just a vulnerability map. It's an illustrative example of a previous work that we did in LA region. The vulnerable areas are red, which isn't very encouraging, but there we go. But understanding vulnerability is going to help with basin planning, so identifying streams for protection, so high quality streams, or even streams of outstanding natural resource waters to be identified or for those, as well as areas of concern if they're highly vulnerable, or where the best areas are most appropriate, or what we think are going to be the most successful areas for restoration. It can also help permitting, so any kind of future projects or proposals that are going to have negative impacts on streams and make them more vulnerable, we can flag those, and that's going to help with protective regulations. So our task was basically to evaluate hydrologic vulnerability for all of the streams in the San Diego region. And just as for the record, our vulnerability, we define that as a change in condition from current to future. But I'm going to talk more about that later on in the talk. And we did this through two different analyses. First of all, we use bioassessment indices, so the CSCI and the ASCII. And that's an ongoing piece of work. I'm not going to talk about that today, but I just wanted to let you know that we're doing that too. But I am going to talk about the higher trophic level species analysis that we're doing, which we're using Arroyo toad as a model species. So the Arroyo toad is a really good kind of model species to use. It represents a higher trophic level aquatic life use, so in terms of beneficial uses. It's already a species of management interest or management concern. Its population has declined rather a lot over the last 20 to 30 years. And it's very sensitive to hydrologic change, so it's very dependent on breeding pools for reproduction, seasonal pools, and it's very particular about what that habitat is like as well. So it has to be quite shallow, slow moving, and sandy substrate, usually in the stream margins. And on the plus side, there is also an existing model built by USGS. Some of the work has already been done, but we can build from that to kind of answer the question. So I'm going to talk about how we do that. So before we go into kind of all the details, I'm just going to give an overview of how our approach fits together. So the approach is basically a species distribution model, which relies on spatial correlations between the toad and its environment. So what we do first is link the toad with landscape characteristics, and that's what we're taking from the original USGS model. And it does that through the known observations of the toad. and links it to the characteristics in the same geographical location. Now, because we want to look at hydrologic change, we are adding in metrics that describe flow. And then we apply a random forest model, which is basically our SDM, or species distribution model, and that produces a probability of occurrence for both current and the future. And then the difference between the two is how we're defining vulnerability. So the figure on the right is an example output from the original model. So it's a probability of occurrence, and it's based on the landscape variables, so topography, geomorphology, soil, and as well as remote sensing data describing brightness, greenness, and wetness. For that, we only have current conditions, so we assume that they're going to pretty much stay the same. With our flow variables, because we want to look at change over time, we have current and future variables. And for the flow metrics, we're using the functional flows metrics, which if you went to the workshop yesterday, you'll probably recognize this and know all about it already. But for those that didn't, they're basically a way to quantify seasonal flow components. So as is outlined in the figure there, so it's peak magnitude, spring recession, and dry season low flows, things like that. And individual characteristics of those flow components, like the timing and the duration, they make up the actual flow metrics, and they're all outlined in the table there. So all these metrics have been identified for California and are supportive of key ecological functions in California as well. So using these metrics, we're basically able to isolate the important flow components or seasons that are important for the Arroyo toad in this setting. Oh, and I should say that all the flow modeling was done by my colleague, Chris Taniguchi-Kwan. I'm not going to go into that today, but yeah, we can answer any questions that you have if you're curious. So this is the output of the model. This is probability of occurrence. I do have a full disclaimer that this isn't the finished result, so it is preliminary. The model's undergoing some refinement still, so yeah, the next couple of slides take with a pinch of salt, basically. But the different colors in the reaches, I hope you can see that. No, it's not so clear, so apologies. But they represent the probability of occurrence from the model, and then the circles represent the actual toad observations. So we have an accuracy of like 81%, which is pretty good. So we're in the where we have the toad observations, we have a high probability of occurrence, which is pretty good. And I should mention as well that this is an integrative probability of occurrence that covers the whole period of record, 2000 to 2014. So as well as the probability of occurrence, we also looked into how important flow is. So we looked at the variable importance. And so this is what you can see on the right there. So 3 out of the 5 most important metrics were flow related, and the highest ones being the peak metrics. Again, this is subject to change, just to let you know. Basically, that means that the model has improved by adding in these flow metrics, and the flow metrics is also very important in the distribution of the toad. But also, like catchment and topography are also really important too. So to assess the future conditions, we are creating climate scenarios that are based on changes in temperature and changes in precipitation. This is all done from a stochastic modeling approach, which, again, I'm not going to go into, but that's been worked on by Chris again and our friends at San Diego State University. But we basically have about 25 of these different scenarios, and we can predict probability of occurrence under each one of these scenarios and then compare that back to our current or baseline model. And then we can use that information to estimate vulnerability. So how we do that is basically we're going to end up with a range of changes in probability. Some are going to be higher, some lower, some are not going to change that much. And we're going to use the direction to basically categorize the vulnerability. So, if a reach or a site decreases in probability, that would be classed as vulnerable. If it increases, it would be resilient, and if it doesn't change much, we're just going to categorize that as limited change. So we can look at that kind of as an overall, which is what it's showing on the figure on the right there. So this is a way of looking at proportion of reaches per scenario. So we look into the differences between each of the scenarios in terms of vulnerability. So each one of those pillars is a different scenario, and this is conceptual as well. So we can look at how many reaches or proportion of reaches are deemed as vulnerable under each one of these scenarios, according to the change in precipitation and the change in temperature. So as an example, what we would expect here is that an extreme scenario that has a large increase in the temperature or a large decrease in precipitation, that will be a lot of a large proportion of the reaches will be deemed as vulnerable. But ultimately, what we're going to have, and we're going back to this example that I showed in the beginning, is what we're going to have is a vulnerability map of the entire study region for each one of these scenarios. And we can use that kind of to inform management. So I'm just going to walk you through an example of what it could be used for. So, so the red areas, as I mentioned, are vulnerable. So that means they're vulnerable to long-term change. So we would suggest that any projects that are coming in that are going to make kind of more flow alteration, we would suggest that more management is needed or more analysis before anything can be implemented. If the area is of limited change, then so that means it's limited change long term, so it's kind of okay. But for any near-term alteration might kind of tip it over the edge. So we want to basically protect that and make sure that doesn't happen. So perhaps we have some mitigation that's required there. And in resilient areas, basically we want to keep it that way. So we want to just monitor that and keep an eye on it, right? So I've spoken about the random forest and the SDM. And even though we can predict in future, it's actually still a temporally static prediction because it's one value over a period of record. And big discussions that we're having with our TAC members is that as arroyo toads are vulnerable to dynamic wet-dry cycles in accordance with their life history, we need to somehow kind of get that within the model. So, for example, arroyo toad has a life expectancy of 5 to 7 years. So what happens under a 5-year drought or a 6-year drought or a 7-year drought? The best way to do that is by applying a population viability analysis. It's not within the current scope, but what we are proposing to do is model the population under different terms of drought, so 5, 6, 7, 8, 9 years, and then relate that back to the toad's life history. I think then we'll be able to get much more of a detailed picture about how toads are vulnerable to long-term drought and understand vulnerability overall. And then ultimately what we'd like to do is, obviously we've focused on one species in one region, but we would like to extend that and establish framework for other species and regions. So if you're looking to assess vulnerability where you are, then do let me know. And yeah, thank you very much.
Hi, that's an awesome talk. I'm kind of wondering if you were thinking about using not just arroyo toad, but other kinds of levels of sensitive species for this work in order to inform, I don't know, like the protection strategies in terms of like not just arroyo toad, which is like very sensitive and really not doing well. Maybe you do a different kind of umbrella species or something like that. Yeah, no,
I understand that. We decided through a TAC meeting that we were going to use the arroyo toad because they were kind of it's such a concern. And we wanted to choose kind of something that's representative of the study area and also kind of the most sensitive. So we're getting kind of like the worst picture, I guess. We would like to extend it to other species, but yeah, finding an umbrella species would be a really good idea. Yeah, but we haven't done that yet.
So your model had high percent correct classification, but what did specificity and sensitivity look like?
I don't know off the top of my head, but it's quite high. I did several different metrics, including R-squared and also the AUC and the TSS. But they were all relatively around that number as well. So like 0.8, 0.85.
Sensitivity and specificity?
Yeah.
Okay.
Yeah, there is, yeah.
Yeah, my question was related to that
Okay.
one. Like, I was wondering if you can also predict absences reliably. And I imagine that there's detectability issues around these species, and maybe, I don't know, if like modeling a suite of species, some more detectable than others, could help you model better the one that's less detectable.
Yeah, good question. We do actually have some absences because a lot of the data we got from USGS, and they go to the same places all the time, and they check these. Obviously, it's not as many as we want, but we also used a kind of spatially explicit way to do pseudo absences. So I think it's kind of the best way we can kind of model the absences. But there is kind of, because the toad is so sensitive and so such a concern, that a lot of surveys are done on it. So there's quite a lot of data if you know the right people. Thanks.
Thank you.
I have a question for you, Katie. So just thinking of the management applications of this, and you'd shown the regional water boards and kind of expanding that to other areas of the state. And so I'm thinking from our perspective, we could use that tool for some protective actions, whatever it may be. But do you have any experience with Fish and Wildlife or other agencies that have kind of oversight over this too, and them using that same tool and assessment results, or plans to do that?
Yeah, good question. I don't know if they'll be using it towards the end. They have been involved in the development because they're part of the, sorry, I don't know if you can hear me, hopefully. But they have been a big part of, they're on our TAC meeting and our stakeholder groups, so they are involved in that. So they obviously want to make it as usable as possible, but I don't know any specifics of how they're going to use it. Thank you. Anyone else?
Any other questions? All right, thank you so much, Katie.
Thank you.
Okay, our next talk, we're kind of keeping in the amphibian theme. Umarfarooq, if you could please come down. Hi. He'll be talking about modeling the distribution of the amphibian pathogen that I'm not going to try to pronounce, so we'll leave that up to you, to inform conservation management. And I'll get your slides up.
Thank you for the introduction. My name is Umarfarooq Abdulwahab. I'm a PhD student at Utah State University. Today, I'm going to talk to you about aspects of my PhD dissertation, in which I'm modeling the distribution of the amphibian pathogen, Batrachochytrium dendrobatidis, to inform conservation management. And just to give you some general background, this work is a component of a project in which we aim to develop a data-driven decision support system to identify optimal land use alternatives for protecting species of concern on the Department of Defense and surrounding lands. And to your right is the map of the study area showing the Department of Defense and National Forest lands where our target landscape occur. Now, how are we developing these decision support systems? We are using a software package known as MarkZan. And what MarkZan does, It does this in a systematic manner where it throws grid cells, which we call planning units, over the study landscape. And in this case, we have around 40,000 of those planning units, with each of them having the size of about 200 by 200 meters. And for each of the planning units, Maxan is assigned information on what area of species to conserve and the risk associated with conserving the area. So basically, we tell Maxan to select planning units that would cost the least amount of money to protect certain species at a very minimal cost. And so one of the ways we are estimating risk, especially for one of the most threatened species across the world right now, which is amphibians, is that we are trying to assess how these species are exposed to the risk of diseases. We know across the world that amphibians are in declines, and there's so many reasons that has been hypothesized to be causing some of these declines, including habitat destruction, global warming, invasive species, radiations, overexploitation, pollution, as well as infectious diseases. But today, my focus will be on infectious diseases. How are we estimating the risk of amphibians to this? To your right, you see very cool pictures of some colorful amphibians. Those are my study species. I'm not going to talk about them today, but maybe in subsequent conferences, you'll find me talking about them. Back to diseases. So talking about diseases, one of the very, very deadly diseases that's going around now across the world is chytridiomycosis. Chytridiomycosis is caused by Batrachochytrium dendrobatidis. It's a mouthful, obviously, and that's why simply we say BD. So BD has been postulated to be responsible to cause decline or extinction to well over 200 species, and it can affect up to about 350 species. And what it does is that it affects the skin of these amphibians, thereby causing disruptions in the skin functions, and ultimately the frogs fall sick and die. And so how does BD dynamics works in an aquatic ecosystem? So it starts with the zoospores. Zoospores would have to attach to the skin cells, and then they form cysts, and when those cysts mature, they then form into zoosporangium, and then from this zoosporangium, they burst out open, and more zoospores explode, get released into the environment, and the cycle continues. But well, now I have painted all the gloomy picture of what is happening to amphibians. What are we doing, or what is being done to address the situation? Well, I forgot to mention that at the minute, we do not know exactly how to manage BD in its natural environment. But one fundamental solution that we need to know is where can we find BD? And there's been effort across the world to document where BD occurs. So this is one of the earliest work by Fisher et al. In 2009, where they tried to document where BD occurs on the landscape. And since then, there's been further works to update these data sets. But we all know that we cannot sample everywhere. So what we do is that we build models to then predict to areas where we cannot sample. And that is the map we can see beneath the distribution maps. So what we can see, although these global analyses are very helpful, but we see from the maps that the factors that drive BD distribution can vary across the landscape. And so therefore, there is the need to understand how BD varies at regional scales. And that informs the questions I'm interested in answering, which is that where does BD occur in my study landscape? And what environmental factors are associated with its occurrence, as well as how does BD habitat suitability varies across the landscape? And to kind of give you some general approach to how I'm trying to address these questions. First, we went into the field and collected water samples, took them to the lab for genetic analysis. From the analysis, we were able to document presences and absences of chytrid, and then in association with several environmental factors, including climate, habitat, human, as well as reservoir species, we're able to identify best predictors as well as predict across the landscape where BD occurs. And so this is how we went about sampling the study sites. So we selected some sites across gradients of water body types as well as physioclimatic conditions. And then at each site, we collected 3 samples, making sure that the locations where we collected each sample was 10 meters apart. And we filtered 1 to 5 liters of water and then took it to the lab for genetic analysis. And so we fitted 2 types of models. And given that we were able to collect repeat observations across the study sites, it allowed us to then fit random forest model, which does not require repeat observations, as well as occupancy models that require repeat observations. So one caveat to these 2 models is that one of them, which is random forest, assumes perfect detection. However, the other type of model can help us access or can help us estimate how imperfect our detections were. And of course, both models, which still requires information on occurrences and environmental predictors to then make predictions across the landscape. Here are the results. This is the occurrences of BD across the landscape. But what we can find is that in most parts of the areas, BD does not occur. However, prevalence of BD is high in certain areas like the Vandenberg Air Force Base, Vandenberg Air Force Base and Fort Hunter Liggett. So how do the responses of the predictors, how do they look? Well, first of all, let me mention that these models perform moderately well, so it may be helpful or useful for conservation planning. So from left to right, we have ranked the variables in order of importance in the models. So what we can find is that occurrence of BD increases with increasing precipitation up to about 4 millimeters before it plateaus. And we see that impervious surface also influences the occurrence of BD, where BD occurrence increases with percent impervious surfaces. And it's very important to point out that although the percentage of impervious surface on this landscape is quite small, but a very small change can significantly cause an increase in the occurrence of BD. We also see that with BD increases with vegetation cover in the landscape. We also see that there's low probability of finding BD in some temperatures, but as from 14 degrees Celsius, the probability of finding BD increases. As well as we find BD in the slopes that are high. So how does the partial dependence plots look like for occupancy model? There we see similar patterns, but what we can, information that we can glean from these patterns is that we can also estimate confidence around the predictions. So whilst the predictions for some of the predictors, while the predictions from some of the predictors are tight, others could be quite high, for example, for percent impervious surface, where we see quite a high uncertainty. But in addition to that, we're able to estimate how well or what influences the detection of BD. And we see that as temperatures increases, the detection of BD reduces. Where else on the landscape can we find BD? So these are the mapped predictions from both the random forest and occupancy model. There are similarities, but there's subtle differences. When you look at the map for the random forest, you see there's not quite, so areas that are blue are not as blue as what we have in the occupancy model. And what we think is that if you look at those partial dependence plots that I showed you, occupancy models can only capture linear relationships, whilst random forests can capture non-linear relationships. So we see those stark extremes in the predictions. Having said that, what is the takeaway from our analysis? Well, we found that BD is unevenly distributed across the landscape, but then there is high uncertainty for some predictions. Some water bodies may have low suitability for BD, whereas other areas can be BD hotspots. the amphibian risk to BD varies across both natural and anthropogenic gradients. Model results will serve as one of the measure of risk when identifying planning units for amphibian conservation. And the likely drivers of BD we found on our landscape further reinforce the view that once BD establishes, it may be difficult to address. With that, I would like to thank my advisors and committee at Utah State University, our collaborators at California State University, Monterey Bay, as well as our funders, CEDIP. Thank you, and I will take your questions. Yes.
Hi, great talk. I'm curious if you could tell us a little bit about the spatial scale of your predictor variables and how those were selected. And I'm just wondering, kind of related to that question, how sort of the spatial proximity or effects were considered, and thinking about, maybe sites along the border of your study areas and potential influences there and influences across the stream network and so forth. So just how were the spatial scales and relationships considered in your occupancy models and rainforest models?
Yeah, so we had divided the spatial scale of the study area into like 50 by 50 meters, and then tried to attribute each of the predictors to those scales, and so that we could have finer predictions. Does that answer the question?
I think so. So it sounds like you considered the environmental characteristics at a particular cell. Is there a way to consider what influence the neighboring cells may have had on the potential presence or absence of chytrid?
If I understand your question very well, you're asking if we find BD in a particular cell, what happens around the surrounding cells? Yeah, we also took into cognizance, proximity of presences to absences in our models, but we didn't find any response from that.
Thank you.
Yeah, thank you.
That was a great presentation.
Thank
Thanks. As far as, I'm curious actually how virulent BD is for amphibians, and you mentioned different conservation practices that can be implemented. If you could give any examples of what can be done
to address BD distribution. Yeah, so you see from our models, which was part of what I gave as the implication, we find some predictors, for example, like vegetation, where we see increasing vegetation, probability of finding BD increases with vegetation. But in that case, it's kind of difficult to address because, vegetation might be important for other species, so we won't go and cut off vegetation. So it's mostly what is important is that, we know where they occur, and then we could make some management decisions. So for example, I showed on my map where there's certain areas that are hotspots. So what people are doing now is that you could isolate some infected frogs from those areas, treat them in isolation, and then return them back. Okay, how to address, so.
No, just how deadly is it for the different amphibians also once they contract BD?
How difficult, sorry, can you?
Is there a lot of mortality once an animal's infected?
Yes, so in the literature we find that some species can actually get infected but don't really get sick out of it. And so that was why even in our models we considered, for example, certain species like boreal toad or the African clawed frog, where we call them reservoir species, where they can get the BD, but then they don't cause the spread. So we considered that also in our models and didn't find any response. Does that answer your question?
I have a question back here for you, kind of related to that. How does how does it spread? So I know once it's there, it sounds like it's pretty impossible to manage, but are there potential management actions to keep it within those hot spots and not move to other water bodies?
Yeah, so, areas where it's been, let's say, established that, okay, BD occurs there, what people mostly do is hard to, really manage in the wild. So what people now do is you isolate them and then bring them to the lab or somewhere and then treat them, then return them back to areas that has been tested and known not to be infected.
As far as that moving, it's not typical where it would move to another water body, like some invasive species or something like that.
Yeah, so of course, so like I mentioned in the life cycle of BD, first of all, the zoospores need a host to attach themselves to. So as the cycle continues, it keeps infecting other hosts, and then that continues within the.
Gotcha. Okay. Thanks. Any last questions? All right, seeing none, thank you. All right, and then we're gonna kind of shift gears a little bit. Moving on to Joe Westhouse, if you want to come on down. Joe's going to be talking from State Water Board perspective on the science supporting statewide biostimulation, cyanotoxins, and the biological condition provisions. And I'll get your PowerPoint up.
So yeah, I'm Joe Westhouse. I am from the State Water Board Division of Water Quality and environmental scientist and a co-lead on the biostimulation, cyanotoxins, and biological conditions provisions. Let's see. The policy is going to be addressing eutrophication across multiple water body types, but this phase of the project that we're working on is on wadable streams only, and the current conditions that we're facing are the increased number of impaired water bodies, eutrophic and anoxic conditions, harmful algal blooms and cyanotoxins, an increased threat to human health and domestic animals and wildlife, threats to drinking water, and it's all just getting worse and exacerbated by climate change. For the wadable streams, we'll be establishing a numeric objective with 3 indicators, the first being total nitrogen, second total phosphorus, and third will be benthic chlorophyll A. There are 3 approaches that we'll be using to derive thresholds, and I'll be getting into those approaches further into the presentation, but they all share some attributes, some of which are that they can either be used alone or in combination with one another. There is a nexus between the biostimulation and biological integrity in the streams, and they are contingent upon policy decisions that my team and I are having a lot of fun grappling with to figure out what we want to decide. And that would include our desired level of protection and the level of confidence that we want to have that our thresholds are meeting our project's goals. So the first approach is the stress response approach, and this is broken down into 2 steps to derive the threshold. The first step is to establish the goal of biointegrity that we want to establish, or our level of protection. The second step will be to establish the likelihood of meeting that goal. So for step 1, we use 2 indices. One is the California Stream Condition Index, and the second is the Algal Stream Condition Index, the first index being focused on benthic macroinvertebrate communities, and the second, which is the ASCI, that is focused on algal communities. So when we are establishing our goal, the question that we want to ask ourselves is, what percentile of reference sites indicate an attainably healthy stream? So on this slide, this is the California Stream Condition Index, which is the CSCI, and on the x-axis, we have the various CSCI scores ranging from low and an assigned status of very likely altered. And as we move up the scores up to kind of the 1.4 range, the condition of the stream improves. So the goal that we're looking at, or decision that we're wanting to make, is where do we want to set our threshold for the objectives and how attainable it can be. So for this example, you'll see that there are the percentages towards the top of the histogram here. We're looking at various percentages. These are all just examples. So at the 30th percentile, we can assume that 70% of the reference sites are meeting the goal that we set, which would be the CSCI score that aligns with that 30th percentile, which is 0.86, I believe. Or we can look at a different option, which would be maybe the 10 percentile of reference, which would indicate that 90% of our sites, of the reference sites, are meeting the goal that we're setting with this threshold, which would be the 0.79. For the purposes of this example, though, let's go with the 0.79 approach. And then for the algal stream condition index, it's a very similar situation with the reference sites spread across a distribution. And then we identify the percentile of reference that will give us our score based on the level of protection that we want to have. Do we want it at the 90% or 70% and so forth? So for this example, I opted to go with the 0.86 at the 10th percentile of reference. So we can assume that 90% of the streams, the reference streams, would be meeting the objective that we're going to put in the policy if we were to use this number. So we have our goals set. It's 0.79 for CSCI and then 0.86 for ASCI. And now we move on to the second step, which would be to establish the likelihood of meeting that goal. We do know that there is an inverse relationship between the indicator concentrations. So the higher total nitrogen gets, the chances of our meeting our goal go down, and vice versa. So with this exercise, we want to ask ourselves, if my stress indicator measures X concentration, the chances of meeting my goal are Y. So we have another chart here, and if you notice in the upper right-hand corner, we have our biointegrity goal for the ASCII of 0.86 or greater. And then this chart basically tells you the probability of meeting that goal depending on the concentration of our indicator. So we have total nitrogen, and if we move up the chart, you'll see that we have a 95% chance of meeting our goal if we have a 0.07 concentration of nitrogen. And then going down the gradient, we will see at, say, 80%, we have 0.24, or I'm sorry, probability is 80% if our concentration is at 0.24 milligrams nitrogen. So another policy question that we have to ask is, where do we want to weigh the level of risk of meeting that goal with the attainability for the dischargers to meet it? Similar scenario, but this would be for benthic chlorophyll A. Again, the score or the goal is 0.86 for the ASCII, and then we have our various probability percentages based on the concentration of chlorophyll A. So for example, at 44 milligrams per meter square of chlorophyll A, we have an 80% chance of meeting our goal. So that's the first approach, and again, a lot of policy decisions that can really indicate the thresholds that we want to set. To support that, we have the percentile of reference approach. So we can say, I would say that a lot of us here know that nutrients and biostimulatory substances are necessary for a stream, but it has to be at a certain level before it enters the eutrophic realm. So given that that is something that we have to decipher between the natural conditions versus what's too much, we have this option or tool, which is the percentile of reference approach, which considers the natural background levels of biostimulation in the environment. And again, it uses reference sites to determine where they land with that when we do our assessments. So with this, you see another histogram, and we have the 85th, 90th, and 95th percentile, and this is for total nitrogens in milligram per liter. So what we can see here on these red dots going vertically down the graph is it's a cutoff point. So say at the 95 percentile that you see towards the top, we can assume that 95% of the streams are meeting the goal that we set with a threshold that's associated with that 95% at, what is that? About 0.3 something. Sorry, the screen's a little small for me. And if we move down to say the 90, the concentration is going to get less and less. For For this exercise, we ask ourselves, to what extent do we want the threshold to consider the naturally occurring conditions of the stream? And the higher that we go with our percentile of reference, the greater range of variability we can have with our threshold in considering the natural conditions within the stream. And then the third approach is the change point analysis approach, which observes the effects of biostimulatory indicators on the community level rather than just the species level. So for our biostimulatory indicators, there are certain taxa that are resistant when the total nitrogen, phosphorus, or benthic chlorophyll A are higher. And then some are more sensitive to that type of environment, so those populations would decline. In both scenarios, we find that the species diversity overall will decline, so that's another thing to keep an eye on with the policy or the thresholds we select. This is, again, using the CSCI and the ASCI, which measure the benthic macroinvertebrate communities as well as the algal communities. And it observes the changes in taxa frequency and abundance, which ultimately leads to changes in the functions of the ecosystem. So here's an example of a nutrient-tolerant taxa, which is the Cladophora, which is a soft-body algae, so it would be measured by the algal stream condition index. And what you see here is with the lower concentration of nitrogen, we have an abundance of this taxon. But as we increase in our concentrations, the populations decline. So that tells us that at some point they're becoming sensitive to the biostimulatory conditions. And this chart graphs where that change point in the community is. It says 0.92 milligrams is where that change point is. And then for ephemerella, which is a mayfly, this is used, the CSCI is what is used to do this measurement. And we see kind of the inverse relationship. As the concentration of chlorophyll A increases, the population of the mayfly also decreases, which tells us that this is a indicator sensitive taxon. So therefore, we know that we have an issue in the water. But where does that issue arise? Again, we're identifying that point to be at 73.9 milligrams per square meter of benthic chlorophyll A. So there's a method or tool referred to as the Threshold Indicator Taxa Analysis tool, referred to as Titan, where it looks at all of these change points, change points, and determines where on each species that change point occurs, and it puts it on a plot like you see right here. And then from there, what we do is find the mean concentration for each of the different categories or assemblages within this analysis. So on the left side of the plot there, I think we have the increaser taxa lined out, and with this, they're finding the mean of where the increaser taxa communities change. And then on the other side would be the decreaser taxa. Again, where it changes is the mean concentration or the change point. So when we conduct this analysis, we can do multiple types of assemblages to generate the information that you see on the table on the lower left-hand side. So for our increaser taxa, benthic macroinvertebrate communities would be 71 milligrams per meter squared for benthic chlorophyll A as the mean change point. For diatoms would be 47, and then soft body algae is 26. So we get a range of scores for different communities and taxons. And again, another fun question for us on the policy side is, where do we find that line amid all of this information that we get from these tools? So in summary, we have the biostim conditions policy, and we have 3 approaches, all of which can be used individually or in tandem with one another to support the thresholds that we derive. None of these decisions have been made yet, still in the works, but yeah, we look forward to seeing what comes from them. Lastly, I just want to give a special thank you to Rafi Mazor for helping develop these tools with SQUIRP, and then also my co-lead on the project, Katie Fong, for availing her analysis as I prepped for this presentation. It was very, very helpful. This is the contact information for my team, and the very last bullet is the web page where you can get more information regarding the policy. So, thank you. sorry. There you go. Any questions? Oh, there you go.
Could you talk a little bit more about how those stressor response curves were developed? I missed.
The first ones, or?
Yeah, the first ones.
Let's see.
Yeah, those.
Oh, these ones? Yeah. I had a question about that too, actually, because there's the probabilities being derived, but I could not find the information as to how.
Yeah, so these are actually just simple linear regression models where we're trying to figure out, do you have an ASCII score above 0.86 or not as total nitrogen increases? And it's, we sort of like stretched the y-intercept all the way up to 1 because we kind of were aware that there's other stressors going on in the landscape beyond total nitrogen. So that's why you have the probability being like, no chance of bad scores when nitrogen is 0, even though we know something else could be happening.
Hello. Hi. Thank you. I just had a question, like, why did you choose HASCII over the diatom ASCII, since it now doubles the cost of analysis and soft algae is recommended but not required to be collected as part of the swamp protocol?
Yeah, we definitely are looking at the swamp protocol as part of the implementation plan for this policy, but as for selecting hybrid over the other two options, it was found from what I recall 2 years ago that the hybrid did perform better than the other two types of collection. Yeah. There you go.
So we did models for both the diatom ASCII and the hybrid ASCII, as well as the CSCI. We have the data available. It really helps us figure out how these streams respond to nutrients. I don't think there's anything in this that would intrinsically require people to subsequently sample ASCII or which version of ASCII. Just this way they know what levels of total nitrogen or phosphorus are likely to have an impact on that community, even if they're not measuring it directly.
One towards the back.
Made you walk for it, sorry. So I had a question, where do cyanotoxins come into this? And then also, how do the biological conditions provisions for nutrients and presumably cyanotoxins compare to the human health criteria?
Let's see. So cyanotoxins are definitely a part of the project. They're just not part of the 3 tools for this phase that we're in. Cyanotoxins we're looking at for more of the human health impacts. It'll definitely be prevalent when we're entering lakes. I believe it may be a part of our streams component as well. But again, this presentation was solely on the 3 tools for the nutrient or indicators. And then as for the thresholds that I think you're referencing, which is like the OEHA numbers for like danger, caution, and warning.
So like, for example, there are human health criteria for like nitrate in water. And I was wondering if the criteria that you're developing as part of the biological conditions are more or less stringent than those. So basically, are we going to be looking at an entirely new set of water quality criteria based on these provisions?
Well, I see my supervisor nodding his head. Did you have something to say on that?
The numbers in the for human health are generally well above the numbers for aquatic.
Yeah, so what we're deriving will be basically order of magnitude lower probably than what we have for human health, and they'll be specific for aquatic life beneficial uses.
Thank you, Zane.
Thank you.
Hi, Joe. Thanks. It was a great presentation. I really like the way you laid out the 3 different approaches to determining the threat to kind of figuring out which thresholds you can track back to biological integrity. I'm curious. So we've got, at this point in the policy, we've kind of landed on 3 different parameters: nitrogen, phosphorus, and chlorophyll A. We'll be developing numeric objectives for those parameters. and then you have 3 different models or approach tools for determining thresholds, all based on biological objectives, but each one of those tools will give you a range or more than one threshold. So I know that there's a lot of decisions that need to be made on the policy side, or are you envisioning sort of a master spreadsheet with just every number that's been derived from these 3 different tools for each one of the 3 parameters?
Well, you're speaking my language.
I'm just kind of curious what's the next product that we can anticipate reviewing in this process?
All right. Well, you're speaking my language with spreadsheets. So I've thought about that a lot, actually. We are finally at a phase in the project where we have the tools available. So the next is the analysis and really diving into them. I think there may be some staff recommendations to management underway at the moment. That's been more Katie's realm than mine. But as for me and my analysis of this, I have thought the same thing, like how do we take all of these potential thresholds and do an analysis to determine where that perfect line is. There's a statistician next to me, so he might have an idea, and then my supervisor may as well.
I would just add that, so the website that was linked at the end, you can download a whole bunch of, there's a lot of resources available there. A lot of this science is published in an ecological indicators article, which is open access, so you should be able to find that. There is a website supplement to that has exactly the spreadsheet you're looking for with all the different numbers. And right now, like Joseph and his team are trying to figure out how to put those numbers together. Some of them, like the reference distributions, really help provide like a good sort of, you don't want to set your number much lower than that because that's going to, identify a lot of reference sites as degraded. The response models provide a good upper limit because if you go beyond that, you'll start really risking degradation, and it's really going to be a decision to how to balance these competing goals. It's a job I don't envy.
Yeah, right. Any other questions? Hey, well, thank you very much.
All right, thanks, Joe. Thanks, Rafi. Okay, we have one last but not least talk before we break for lunch. Janelle, if you want to head on down. Janelle's going to be talking about biotic condition assessment in the Carmel River after dam removal and river reroute. And I'll get your slides up.
I'm just going to practice, okay. Hello, everyone. My name is Janelle Vasquez-Nicholas. I'm a graduate student at CSU Monterey Bay, and today I'm going to talk to you about my thesis work. And like mentioned, the name of my presentation is Assessing Biotic Conditions in the Carmel River after River Reroute and Dam Removal. So during this presentation, I'm just going to give you a little background on the project, the study objective for this project, the monitoring that was done, some preliminary results, and then discuss a little bit of what these results may mean. And then we'll have the Q&A at the end. So for anyone that's not familiar with the Carmel River, it's in the Monterey area in the Central Coast. And historically, Carmel River has been the Monterey Peninsula's main water source. Going back to 1880, Hotel Del Monte was opened, and this was kind of one of the largest developments in the area, and it initially brought thousands of visitors during a time when the area was highly undeveloped. And this brought in a need for more water demand. So this led to building the first dam on the Carmel River in 1883. And this dam was about 30 kilometers upstream from the mouth of Carmel River, and this was near where the later on San Clemente Dam was built. So as the area began to develop more, so did the need for water. And, I went 1, 2, okay, there we go. So did the need for water in the area, and this led to building a larger dam, the San Clemente Dam. This one was built in 1921, and then when that one was enough, they built another dam, and that was the Los Padres Dam in 1949. And so I'm going to be talking about the San Clemente Dam. That one was the one that was removed a few years ago. And the San Clemente Dam was a large 32-meter high concrete arch dam, and it initially held about 1.76 million cubic meters of water. So after San Clemente Dam was built, it began to decline for many reasons. One of the main reasons is that it accumulated a lot of sediment. Near the end of its life, it contained about 1.91 million cubic meters of sediment. And also in the early 1990s, it was determined to not be seismically safe. So that meant in a large flood or a large earthquake, the dam could collapse. And then also again, there are two threatened species that use the river, and that's the South Central California coast steelhead trout and the California red-legged frog. So here we have a picture where I wanted to show you kind of like the sediment. In 1994, you could see when there was more water in the river, there's some evidence of sediment down below in the red circle. But then in 2002, when the river wasn't as filled with so much water, you can really see the extent of that sediment that was accumulating in the Carmel River. So all of these issues led to the removal of San Clemente Dam in 2015. And one of those issues was the sediment problem. What do we do with all this sediment if we're going to remove the dam? So a way around that was to reroute the Carmel River. So in 2015, this was the largest dam removal in California, but this is the first time that river reroute was used as a strategy to prevent large amounts of sediment from moving downstream. And so if you look at the figure, on the right-hand side, we have where the Carmel River initially flowed. And then on the left-hand side, we have where the Carmel River was rerouted. And so the Carmel River was rerouted into the San Clemente Creek, which was useful because the San Clemente Creek ran parallel with the Carmel River before their confluence, and the dam was built right beneath their confluence. So that kind of gave the engineers a way to reroute the river. And in addition to that, because the Carmel wasn't going to run on the right-hand side anymore, it left an area where they can stockpile the sediment instead of having to haul it out of the site. So this was called the Carmel River Reroute and Dam Removal Project. And so the river reroute also included engineered habitats that could be used by the steelhead trout to increase their habitat and also their passage. And like I mentioned, sediment was sequestered on site. And in addition, there was a large native plant restoration that was done. So this project had different intended benefits. The first is that it eliminated those safety concerns, the seismic flooding and erosion concerns. And it also had the benefit of increasing habitat for steelhead trout, especially in an area where they haven't really been able to pass that dam in about 100 years. And then additionally to that, it would improve sediment transport and woody debris transport naturally. And then there were other benefits like reducing competition for the California red-legged frog and also increasing benthic macroinvertebrate diversity, which we know is a food source for steelhead trout. After the dam was removed, there were some big challenges that happened. Specifically in 2017, there was 3 major floods. There was a 30-year flood and 2 10-year floods. And that kind of destroyed all the engineered habitat that they did and moved some sediment downstream from the area. And then in 2019, there was boulders that were moved as well during some other flooding events. So based on this, this project kind of wanted to understand what are the biological conditions in the river, given that this is the first big dam removal in the state, but also it's the first one to use river reroute. So this work is part of a 5-year monitoring program. And specifically, I'm looking at the benthic macroinvertebrate aspect to that. And so I had 2 hypotheses. The first is that the BMI their community composition would improve downstream and upstream of the dam compared to after another study that was done after the dam was removed. And also downstream would be more ecologically intact compared to the upstream area where the river was rerouted. And so in 2018, McCann et al., They did the first BMI monitoring, and they used swamps bioassessment. They subsampled those the invertebrates, and then they used the CSCI to look at the condition in the river. And then all of that was done again in 2022. And so here we have our study sites. The orange is the downstream area, and that I'm going to call downstream for the results. And then the blue is in the rerouted section of the river, and I'm going to refer to that as reroute. On the left-hand side, we have standing in the reroute looking upstream, and then on the right-hand side, standing in the downstream section looking upstream. So we have some preliminary results. I'm about 80% done identifying my invertebrates, so these are just preliminary. But already we can see that the reroute has a much lower CSCI score compared to the downstream. It actually moved from possibly altered to likely altered. And while the downstream does have a slightly smaller CSCI score, it's still within the same threshold as before. And then looking at the metrics individually, we can see that there's a loss of sensitive taxa in the EPT, shredder, and tolerant taxa for both of the sites. Here we have a table with the taxa that had a capture probability of at least 50%. And what I wanted to focus on is riffle beetles weren't found in 2018 or in 2022. We have a loss of some taxa, lepidostoma and microcema, either in both sides or in one site. And then we have taxa that wasn't seen in 2018 that we did see in both sides so far. Here we have a table with some of the physical and chemical properties that were taken. And what I wanted to focus on here was we saw an increased presence of macroalgae and macrophyte. And then while the canopy cover did improve for both sites, we can see that the downstream still has a much larger canopy cover than the reroute. So why do we have these results? Some thoughts that we have is that the sites are being impacted by a recent fire that occurred in 2020. I don't know if you guys saw the poster by Yokomizo and Olson. They've been studying the increase in phosphates in the river after the fire. Additionally, Smith et al. In 2021, they found that gravels did not replenish in the area. It was estimated that the debris flow that occurred after the fire would input more gravels in the area, which are really needed, but that didn't happen. And then additionally, the Los Padres Dam upstream is kind of preventing some gravel input into our area. Additionally, we're going into an additional year of drought, and maybe that's could be impacting the loss of taxa that we have. And then as I said earlier, I think that between the reroute and the downstream, that lack of canopy cover is impacting how the taxa can withstand some of these changes compared to the downstream. And I wanted to thank my thesis committee members, Dr. Doug Smith at CSU Monterey Bay and Keith Wright at AECOM. AECOM is also funding this project, so I wanted to thank them for that. And all the students that helped me in the lab, in the field, I couldn't have done it without them. And with that, any questions?
So the 2018 and 2022 water years were really different out there. Did you guys look at the hydrographs and kind of compare the difference in flow habitat structure through the season up to the point where you sampled the benthic macroinvertebrate?
Yeah, so I just looked at the yearly flow in the area so far. And 2018 was right before a high water year in 2019. And then 2020, 2021 is like it's like very low. So definitely I think that there's that big difference in the flow in the rivers. But yeah, I haven't really done any data analysis with that.
I really like that you looked at the specific taxa that changed between 2018 and 2022. I'm wondering if you have any thoughts about the type of life history traits that might be driving some of those changes.
Not yet. I did notice though, I know oligochaetes, they are definitely more tolerant, and I think that kind of tells us that some of the conditions may have changed for them to, I saw quite a bit actually. And then the reroute had more than half chironomids. So I think that's kind of something to look at as well. I forgot to mention that both sites had more macroalgae, and I think that could be due maybe to possibly increase nutrients, especially because both of them have more canopy cover. So I don't think more light's doing that. But yeah, I'm definitely going to take a deep dive into that.
Hi, Janelle. I have one question too. As far as follow-up, so it sounds like this was a highly managed system. They did some rerouting and restoration, stuff happened, and we see kind of a decrease in CSTI. And it seems to me like that data could be really useful to whichever agency or whoever did this restoration work to maybe come back and augment gravel or whatever it may be. So the question is there a feedback loop or some way, are you working with those folks that did the original restoration work, or, yeah?
So yeah, we're being subcontracted by AECOM. They're the ones that are doing the monitoring right now. I think basically what they want to know is what is the condition of the river, because they did do all that restoration work. And I think they're hoping that whatever the results are, they can help manage it, whether it needs more management or, yeah, if what they did was enough.
Cool. Good to see your data being put to use. All right. Any other questions for Janelle? Oh.
Yeah, I was just curious if you looked at the volcanism and if you saw any change in that.
I have not yet. I'm still in the process of finishing up my invertebrates, but that's definitely something I want to look at.
Any last questions? All right. Thank you so much, Janelle.